Good evening. For our non-scientific audience presentation, which includes the members Payton Emerick, Adam Laken, Lily Jiang, and Maya Genge, we decided to do the scientific paper about microplastic prevalence in Anatolian water frogs. You can switch to slides. So for the introduction for this scientific paper, basically the research we're looking at, microplastics, which I know plastics. Plastics are an incredibly useful material that can be used for many everyday products, which is why global plastic production has increased dramatically since the 1950s. Basically, since the 1950s, I mean, you can see it everywhere you go. Plastic products are pretty much everywhere, from toothbrushes to just things that you use in your everyday life. But when you throw and so an annual output has reached 368 million tons in 2019, and plastic production is continuing to increase. And predictions say that by 2050, the expected total output will be 50 billion tons, which is a lot of plastic. However, the problem with using plastic materials is that plastic can be broken down into millions of microplastic particles. And basically what a microplastic particle is, it's just a little plastic fragment that's less than 5 millimeters and they're easily dispersed into the atmosphere, aquatic and terrestrial habitats. This raises environmental concerns, and several recent ecotoxicological science studies have revealed that certain organisms may suffer from reproductive problems, oxidative stress, organ injuries, psychological weakening, and eventual death due to the ingestion of microplastics, which could be really bad for the environment, especially looking to the future, because with the increased use of plastics, there will be more microplastics in the environment. So this researchers in this study, they looked at frogs because frogs are an amphibian species that are incredibly good bioindicators. However, populations have declined declined by about 70% due to pollution and other environmental problems. So measuring microplastics amounts in frogs is something that has seldom been done before, which prompted the scientific question: what is the amount of microplastics found in freshwater frogs, and what could this mean for the decline in frog populations? That was their scientific question that allowed them to form the hypothesis that microplastic contaminations found in frogs are a good indicator of the amount of microplastic contamination in a food chain or ecosystem. And this entire hypothesis prompted the experiments that are described in the scientific paper.
Okay, so for assays and methods, there were a total of six assays and methods. And for the first one, it was sampling and microplastic extraction. So first, they got adult marsh frogs and levantine frogs, and they were collected from 19 different stations in Turkey, where they were all naturally distributed. And then the frogs were euthanized and preserved after. And so to find their experiment, the scientists measured the length and the weight of each frog. Their gastrointestinal tracts, or the GITs, were then removed from the frogs, and each GIT was placed in H2O2 and kept for 3 days. Then they were filtered using a vacuum pump, where they were then stored in petri dishes. The second assay and method was contamination control of microplastics. So it was really important to make sure there was no outside contamination of microplastics to make sure this experiment was 100% reliable without any outside sources. So to avoid microplastic contamination of the groups, the researchers used cotton lab coats and polymer-free gloves. All liquids were also filtered through a paper to remove microplastics. And all the labware was washed and covered with aluminum foil to prevent further contamination.
The next method used was the recovery rate of microplastics. This was basically to make sure that all the microplastics that were in a given frog were being recovered. So they took portions of microplastics PET and PE, and they were prepared and ground. They then put these portions into a frog and later recovered them to measure whether or not they were able to extract microplastics successfully out of the frogs. And the recovery rates for these were 97% and 98% respectively. The next method was microscopic examination. Particles that were suspected to be microplastics were transferred to filter papers and classified as fiber, fragment, film, foam, or pellet through examination under a microscope.
After microscopic examination, the next method was polymer verification. The plastic polymer was identified through a random selection of 200 fragments. They were then inspected through infrared analysis. These particles were placed on a reflection crystal and were matched with a reference polymer. Particles that matched this reference polymer by 70% or more were then confirmed to be microplastics. Lastly, data analysis was performed. At every station where the frogs were taken from, microplastic data was defined as a microplastic individual. One test was the Kirkwood-Wallace test that was used to test the variation of microplastics among the sampling sites. Another test was the Spearman collection, and that was used to show the relationship between frog size and abundance of microplastics found within these frogs.
These are the results that the study found. Out of the 176 frogs, microplastics were found in 147 of them. Out of all the isolated particles they obtained, 80% were plastic polymers. Station 9 had the lowest abundance with 0.2 microplastics per frog, while Station 17 had the highest with 18.93 microplastics per frog. And they found a correlation between the population in the city and the abundance of microplastic per frog, as Station 17 was the third most populated city in Turkey. However, no correlation was found between microplastic abundance and the weight and length of the frog. Out of all the microplastics found, 9 different polymer types were found, and 90% of the microplastics were smaller than 300 picometers.
So, how I interpreted these results was that no significant variation in microplastic contamination was recorded among sampling stations, and further research on microplastic accumulation in several other species has shown that there's no significant link between microplastic accumulation and height and weight measurements. So, although they did find a significant amount of microplastics in the frogs, it wasn't found to have a correlation with their weight or their length. And microplastic size is more critical than polymer type for aquatic organisms. Microplastic size and retention times are negatively correlated, meaning that if the size decreases, the retention will last longer, and vice versa. The results of this study were not able to show clear evidence of microplastics impacting the weight size of the frogs, further concluding that the impacts of microplastics on biological forms are still unknown. And decreased synthetic fabric manufacture will thereby drastically lower microplastic emissions into the environment. Overall, I agree with these interpretations because within the study there were graphs and visible results that support that there is no clear correlation between the microplastics and the frog's length or size.
For the conclusion, the paper was able to conclude that presence of microplastics were remarkable at every sampling station used within this experiment, although they were not able to conclude a relationship between frog size and microplastic abundance. Variations in microplastic abundance based on location could be due to differences of exposure at different sampling locations. Microplastics are present within frogs, which do make up a large proportion of amphibians, but more research is needed to reveal potential negative impacts if there are any present. Microplastic presence within frogs is significant as amphibians have been used as biological indicators to monitor environmental pollution in the past. Microplastics are found in other organisms as well. The frogs may have obtained microplastics by ingesting plastic pollution or by eating prey that contains microplastic within their bloodstream. This would be of concern for human health as our processed food is exposed to a lot of plastics, but also animals eat each other that we then eat, and they could acquire microplastics from the food chain. For future studies to understand the negative impacts of microplastics on various organisms' health, including reproductive health, growth, and mortality, are needed to develop a true understanding of microplastic harm. More studies to localize higher incidence of microplastics in frogs are also beneficial as they are used as an environmental pollutant indicator. So in places like Turkey or the United States, we can kind of gauge the microplastic pollution based on the amount of microplastics within these animals. I think it would also be beneficial to develop methods to test microplastic levels without euthanation and GI tract isolation to measure this amount in other organisms, as we are not able to do this in larger organisms without ethical concerns like humans or cows. Studies on microplastic levels throughout organisms in a food chain and their potential cycle back to the environment is also necessary to understand how in depth they are and how much they can travel throughout an ecosystem. And then this is our source.